Welcome back to another exciting set of video notes. Today we're going to dive into the topic of enzymes. Remember, enzymes are specialized proteins that catalyze chemical reactions. All right, let's make it make sense. By the end of this video, you should be able to understand the function of enzymes, appreciate the specificity of enzymes, and analyze how environmental conditions influence enzyme activity. Okay, let's start off by understanding what exactly are enzymes. Enzymes are like the superheroes of biology. They're specialized proteins that speed up chemical reactions in living organisms. Imagine a reaction as a boulder at the top of a hill. To get the boulder rolling, you need to give it a push. Well, in chemical reactions, in order to start a reaction, you have to provide what's called activation energy. And enzymes will swoop in and lower the activation energy required to start the reaction. This essentially makes it happen faster and more efficiently. Activation energy is one of your vocab words, and it's the energy required to start a chemical reaction. So remember, enzymes lower that amount of energy. A chemical reaction occurs when bonds are broken and or reformed. Let's use the chemical equation for photosynthesis here to help you guys understand this concept. Here, you can see that the equation states 6CO2 plus 6H2O yields, anytime we see an arrow like this, we're going to say yields, C6H12O6 plus 6O2. Another way to read that would be 6 carbon dioxide plus 6 water makes 6 glucose molecules and 12 oxygen. In this equation, carbon dioxide, CO2, and water, H2O, are our reactants. They come together with the help of sunlight and an enzyme called rubisco to form glucose, C6H12O6, and oxygen. These are the products. To really understand this, let's focus for a minute on the equation. On the left side of the equation, you have the reactants, carbon dioxide and water. These molecules contain high energy bonds. To convert them into glucose and oxygen on the right side of the equation, those bonds need to be broken and new ones need to be formed. This process requires energy input, just like it would in any chemical reaction. This is where enzymes come into play. Enzymes are biological catalysts, and their role is to speed up chemical reactions without being consumed or changed in the process. In the case here of photosynthesis, enzymes facilitate the conversion of reactants, CO2 and water, H2O, into products, glucose and oxygen. And they do that by lowering the activation energy required for the reaction to occur. Let's visualize the energy needed for photosynthesis with and without enzymes using a graph. As you can see, the reactants, our substrate, and our products are the same with or without enzyme. The blue line is indicating what it looks like with enzyme, red is without enzyme. The graph here shows that energy needed for photosynthesis to take place with an enzyme, activation energy with an enzyme, is lower than without an enzyme. Enzymes significantly reduce the amount of energy needed, also known as our activation energy, making the reactions more efficient. We'll be looking at this graph later on in class as well. Next, I do want to discuss how specific enzymes are. We call this enzyme specificity, and we have a few key terms here, so please write them down. A substrate is the molecule that enzymes act on, and enzymes have an active site, which is a specific region where the substrate fits perfectly. Imagine the active site of an enzyme as a lock and the substrate as the corresponding key. Just as a specific key fits perfectly into a lock, only the correct substrate can fit precisely into the active site of an enzyme. This ensures that the enzyme only catalyzes the intended chemical reaction by binding with the appropriate substrate. This high degree of specificity is essential for the efficiency and accuracy of reactions in living things. It prevents unwanted reactions and ensures that the right reactions occur at the right time and the right place, which contributes to the overall regulation and control of biological processes. All right, transitioning from the lock and key model, let's understand the concept of an enzyme substrate complex. When substrates bind to the enzyme's active site, it forms the enzyme substrate complex. This complex is a temporary union. While in the enzyme substrate complex, the enzyme facilitates the conversion of the substrate into products. It does so by either bringing the reactants closer together or providing an alternative reaction pathway that requires less energy. Once the reaction is complete, the enzyme substrate complex releases the products of the reaction. It's important to note that the enzyme itself remains unchanged and is not consumed in this process. Write that down, please. It's free to bind with new substrate molecules and catalyze additional reactions. Enzymes are highly efficient because they can be used repeatedly. After releasing the products, the enzyme is available to bind with more substrate molecules and continue catalyzing reactions. All right, moving on to optimal ranges. Enzymes are known to be picky, and they work most effectively within a specific range of conditions. We're going to refer to the range in which they work best as their optimal range. Exposure to conditions outside of their optimal range can make enzymes less effective or even denatured. Remember, denatured means that enzymes lose their shape, and that can make them non functional. Right. Factors like pH and temperature can influence enzyme activity. We're going to call those our environmental factors. And the optimal ranges do vary depending on the individual enzyme. But in regard to pH, if the pH becomes too acidic, which would be a low pH, or too alkaline, which would be a high pH, the enzyme's active site and overall structure can be altered. This can result in the loss of the enzyme's shape, making it unable to properly interact with the substrate, which would prevent it from catalyzing reactions. Like pH, enzymes also have an optimal temperature range at which they work best. High temperatures can cause the enzyme molecules to vibrate vigorously, leading to the disruption of chemical bonds and interactions that maintain the three-dimensional shape. That's often irreversible and results in denaturing. Low temperatures can slow down enzyme activity, but typically don't denature the enzyme. Instead, they reduce the rate of enzyme substrate collisions, the rate of the reaction. All right, now let's discuss concentration. Enzyme concentration refers to the amount of enzymes present in a given area. The concentration of enzymes directly affects the rate at which reactions occur. The fundamental principle here is that higher enzyme concentrations result in faster reaction rates. Why? Because with more enzymes available, there are more active sites ready to bind with substrates and catalyze reactions. Even at relatively low concentrations, enzymes can cause reactions to happen. However, as you increase the concentration of enzymes, the reaction rate typically increases. It is essential to note, so please jot this down, that increasing enzyme concentration can only speed up a reaction to a certain point. Eventually, a maximum reaction rate is reached when all available substrate molecules are bound to enzymes, and the reaction cannot go any faster, unless, of course, we add more substrate to bind to those available enzymes. All right, that brings us to inhibitors. We do have two types. We have competitive and non-competitive inhibitors. And an inhibitor is going to be a molecule that binds to an enzyme, decreasing its activity. It's inhibiting the activity. All right, let's start with competitive inhibitors. Competitive inhibitors are molecules that closely resemble the substrate required for the reaction to occur. These inhibitors compete with the substrate for access to the enzyme's active site. And they do that by binding to the active site of the enzyme, essentially blocking it from the substrate. Since the inhibitor and substrate cannot occupy the active site at the same time, the enzyme's function is hindered. It cannot work. In most cases, competitive inhibition is reversible. When the competitive inhibitor is no longer present, the enzyme can resume its normal activity by binding with the substrate. Some medications actually work this way. So, for instance, certain prescription drugs will mimic the structure of molecules in the body and compete with them for enzyme binding sites. All right, alternatively, we have non competitive inhibitors. These, on the other hand, do not directly compete with the substrate. Instead, they bind to a different site on the enzyme. We call that the allosteric site. This binding causes a change in the enzyme structure, typically altering the shape of the active site. If we change the active site's shape, it makes it less effective or even completely unavailable for substrate binding. As a result, the enzyme cannot catalyze the reaction as efficiently or often at all. Non competitive inhibition can be reversible or irreversible. It depends on the specific inhibitor. So, some non competitive inhibitors bind tightly and permanently alter the enzyme, while others can be displaced or knocked out by the competitive inhibitors or even a substrate. All right, lastly, let's talk about the naming conventions of enzymes. Enzymes are often named because of their substrate or the type of reaction they catalyze. The ASE ending in many enzyme names is a common indicator you're dealing with an enzyme. However, it's important to note that not all enzymes follow this naming convention. Enzymes do often provide clues about their function. For example, the enzyme lipase is involved in the breakdown of lipids or fats, while amylase is involved in the digestion of amylose, which is a starch. Some enzymes, though, as I said, don't follow the ASE naming convention. For example, we have trypsin here, chymotrypsin. There's an enzyme pepsin, which is involved in protein digestion. It does not end in the ASE ending. So it is a good hint, but it doesn't always work that way. All right. In summary, remember these 5 things. Enzymes are specific. We have the lock and key model. They're efficient. They catalyze or speed up chemical reactions. They're picky. They have optimal pH and temperature ranges. They're breakable. They can be denatured. And they're blockable because inhibitors can cause them to not work. Okay, congratulations. You finished Unit 2 video notes. Remember, we'll be having mastery checks in class for this unit. So if you have any questions or need help, please come visit me down at the tutoring center. All right, I'll see you in class.